Hello students, welcome to Saurav's classes. My name is Ashmita and in today's class we are going to talk about the latest updated syllabus of WBCS Mains optional paper of Botany, and in today's class we are going to discuss about the syllabus of paper 2. Hopefully the candidates who are choosing these optional papers, that's mandatory for all of you, and if you are choosing these papers, you would know that you have to attempt two optional papers of the same subject. Whatever subject you are choosing, you have to attempt two optional papers on that. So here in today's class we are going to discuss about the syllabus of paper 2 of Botany. In our previous class we have already discussed the latest syllabus of paper 1 optional paper of Botany. In today's class we are going to start discussing about paper 2 of Botany optional paper. So let's start our class. Let us start with the discussion of paper 2 syllabus. Paper 2 starts with cell and molecular biology. Here you will get cell biology, cell and its types, prokaryotic and eukaryotic, structure and functions of the major cell organelles, then cell division, the significance of cell division, then cell cycle, structure of a typical chromosome, nucleosome, then model of the chromosome, chromosomal aberrations, then concept of the RNA world. Then there is this section on molecular biology, having chemical structure and nature of the nucleic acids, concept of the gene, replication of DNA, concept of genomic DNA and cDNA, then split genes, overlapping genes, oncogenes, genetic code, protein synthesis in prokaryotes and eukaryotes, then central dogma, basic of recombinant DNA technology. Next section is based on genetics, and here you have Mendelian principles, the concept of linkage and crossing over, then polyploidy, multiple alleles, point mutation, sex-linked inheritance, cytoplasmic inheritance, basic population genetics. Then we have the section on plant breeding. Here you have objective of plant breeding, then methods of propagation in relation to breeding methods, then methods of plant breeding, hybridization concept and causes of heterosis, then maintenance of germplasm, heterosis and hybrid seed production, then molecular breeding, maintenance of germplasm, mass selection and pure line selection, heterosis and hybrid seed production, and molecular breeding. Then we have biometry, where there is random sampling, frequency distribution, central tendency, arithmetic mean, median and mode. Then measurement of dispersion, standard deviation, standard error of mean, test of significance, t-test, chi-square test for goodness of fit, then probability, measurement of gene frequency, overview of bioinformatics, the nature of biological data, literature databases, then introduction and overview of biological databases, nucleic acid sequence databases, then GenBank protein sequence databases, introduction to BLAST series. Then we have this evolution biology. Here you are getting questions based on concept of biological evolution, evidence of organic evolution, then Lamarckism, then Darwinism and mutation theories of De Vries. Next you have the section on physiology and biochemistry. Here you have plant-water relations, stomatal physiology, mechanism of opening and closing of stomata, then organic translocation photosynthesis, photochemical reaction centers, then cyclic and non-cyclic electron transport, water splitting mechanism, photophosphorylation, Z scheme, Calvin cycle, biochemical reactions and stoichiometry, photosynthetic efficiency of C3 and C4 plants and crop productivity, photorespiration, crassulacean acid metabolism, respiration EMP pathway, TCA cycle, ETS and oxidative phosphorylation, then oxidative pentose phosphate pathway and its significance, then beta oxidation of fatty acids and significance, nitrogen metabolism is there, then structure and function of denitrogenase complex, ETS of denitrogenase, the basic concept of NIF and NOD genes, then plant regrowth regulators, then photoperiodism and plant types, phytochrome, vernalization, then concept of biological clock and biorhythm, then seed dormancy, physiology of senescence and aging, then stress physiology, biochemistry as a molecular logic of living organisms, axioms of living organisms, then major compounds of living beings, pH, buffers, and basic bioenergetics, chemical structure and properties of the water molecule, then ionization of water, Henderson-Hasselbalch equation, titration curve, and the concept of preparation of any buffer solution, and biomolecules, then general structure, properties, classification, and metabolic importance of carbohydrates, proteins, lipids, and nucleic acids, enzymes, basic structure, nomenclature and classification of enzymes according to IUBMB, then mechanism of enzyme action, then the concept of the active site of an enzyme, activation of free energy, principles of enzyme action, then enzyme kinetics, reversible and irreversible enzyme inhibition. Then you have this allosteric enzyme regulation and covalently modulated enzyme regulation. Then the basic concept of ribozymes, abzymes, and isozymes. Then you have this section on pharmacognosy. Pharmacognosy and its importance in modern medicine. You have to learn and go. Then crude drugs, drug evaluation, secondary metabolites, interrelationship of basic metabolic pathways with secondary metabolite biosynthesis with special reference to Syncona, Ipecac, Athatoda, and Curcuma longa. Then we have this plant biotechnology and instrumentation, plant tissue culture and micropropagation, plant genetic engineering, brief concept of different gene transfer modes and methods, transgenic plants, principles and applications of simple, compound, confocal, and electron microscopy, colorimetry, visible and UV visible spectrophotometry, differential centrifugation, PCR, RT-PCR, gel electrophoresis, blotting, and ELISA. So that was all for this latest syllabus of botany optional paper, and that was the entire discussion. If you need the guidance of a good mentor and a good coach for better preparation, then you can join Saurabh's classes. In Saurabh's classes, we provide you with live classes, regular lectures, online, offline classes, doubt clearing sessions, then revision classes, mock test facilities, crash course facilities, and we also solve the previous year's question papers. We also solve the unsolved papers and also make you practice the sample papers. So if you want to get all these benefits, then you can join our classes. If you get enrolled to our classes, we are going to provide you with the recorded lectures portal access. That is basically those are the live lectures which we record and upload it in our portal, so that enrolled candidates can check those lectures again and again if they want to, and if they face any kind of confusion, they can definitely go through those lectures again and again. It's the portal is available to the enrolled candidates 24/7. So if you want to enroll in our classes for any kind of competitive examination or any kind of entrance examination, then you just need to call us. Number is given in the description box below, and you can just give us a call. And if you have any kind of doubts regarding any kind of competitive examinations or any kind of entrance related examinations, then you can ask us about those examinations. We will clear all your doubts, and if you want to enroll with us, then definitely you can do so. And if you want to know more about our classes, you can check Saurabh's classes website. The website link is also given in the description box below. So that was all about our classes, and that was all for today's class also. We are going to meet once again in our next classes.